Good evening everyone, we are from Group 1 consisting of 7 members. Today we are presenting the main principles of biochemistry that Ligno Biotech applies in processing or manufacturing their product. Several species of tiger milk mushrooms, for example Lignosus rhinoceros, Lignosus cameronensis, Lignosus tigris. First, let's focus on the cultivation of tiger milk mushroom sclerotia. Common substrates include rice or other grains. For example, Lignosus rhinoceros is cultivated using a substrate comprising sawdust mixed with gypsum and rice bran. The cultivation of L. rhinoceros involves the following steps: inoculating a sterile substrate in a container with mushroom spawn. This ensures proper sterilization to prevent contamination. Incubating the inoculated substrate in darkness at a temperature between 25 to 32 degrees Celsius for 3 to 4 weeks to generate mycelium. Providing a casing soil composition to cultivate L. rhinoceros. Next, burying the substrate 2 cm to 6.5 cm under the casing soil after incubation and incubating the buried substrate at temperature between 28 to 32 degrees Celsius with moisture content of 50 to 70%. There are some biochemical principles in the cultivation of tiger milk mushroom sclerotia. First, substrate composition. The selection of a suitable substrate such as sawdust mixed with gypsum and rice bran provides essential nutrients for the growth of tiger milk mushroom. Breakdown of complex organic compounds into simpler forms occur during substrate utilization by the mushroom mycelium. Inoculation and mycelium growth. Inoculating the sterilized substrate with mushroom spawn initiates the growth of mycelium. Enzymatic reactions are involved in the colonization of the substrate by the mycelium. Temperature and moisture regulation. The specified temperature range and moisture content during the incubation are crucial for biochemical activities. Enzymatic reactions, metabolic processes, and the regulation of water content influence mycelial growth and overall mushroom development. Casing soil composition. Providing a casing soil composition involves selecting materials that support the mushroom's fruiting stage, which will contribute to the initiation of the reproductive phase. Understanding these biochemical principles is crucial for optimizing the cultivation process, ensuring the production of tiger milk mushroom sclerotia with desired characteristics and bioactive compounds.
Next, we move on to the next step, which is harvesting the mature mushroom. How do we know it's already mature and ready to be harvested? Is by looking at the structure of the sclerotia. Mature sclerotia are firm and compact masses. When harvesting, there is always a debate about the best way to do it, whether by picking or cutting. But studies have shown that it makes little difference to the mushroom mycelium. Especially when it comes to cultivated mushrooms, we can harvest it either way as long as we remember to handle it gently and carefully. And always remember to use clean tools or knives so that we can avoid contamination on our mushroom. Then, after harvesting, come the next step, which is cleaning and drying. Mushrooms or sclerotia that we harvest usually have dirt and debris that's often attached to the base of the mushroom stem. So, in this step, the mushrooms have to be cleaned thoroughly by using brush or dry paper towel, or we can just run them under cold water to remove all those debris or any residual substrate. Then, dry the clean sclerotia using methods such as air drying or low temperature drying in the hot air dryer. The machine dries the mushroom using the circulating hot air, which the temperature can be controlled depending on the wetness of the mushrooms. The low temperature is very important as it helps preserve the active components in the mushrooms that is sensitive to high heat. It also helps in preventing the spoilage. Okay, next we have milling and the grinding of the tiger milk mushroom sclerotia. This process involves the grinding of dry mushrooms to mushroom granules and then into mushrooms powder by using certain machines like coarse crusher and pulverizer. The supplement products of Ligno tiger milk mushrooms usually comes in powder form because aside it's more cheaper than the capsule form, it also have faster absorption rates in human body and also contain more nutrients, making them a good source of B vitamins and proteins.
There is 3 varieties of this mushroom, and and their size of pores. First one, the Lignosus rhinoceros. The size is 6 to 8 pores per millimeter. Second, Lignosus cameronensis. The size is 2 to 3 pores per millimeter. And the last one is Lignosus tigris, 0.5 to 1 pores per millimeter. Next, quality control. First, safe and hygienic. Implement rigorous quality control measures to ensure the purity, potency and safety of the sclerotia powder. Conduct testing for contaminants such as heavy metals, pesticide and microbial agents. Second, the sclerotium is harvested at the optimal condition prior to the formation of cap and stem, ensuring highest medical value. Third, standardization of production batches to ensure the consistent quality. Standardize the production process to ensure consistency in the concentration of active components in the final product. Monitor and adjust processing parameter as needed to meet quality standard. Fourth, can be successfully scaled up to ensure sustainability. Fifth, safety, quality, and efficacy are scientifically validated. Ensure compliance with local and international regulation related to food safety, quality, and labeling. Obtain necessary certification such as Good Manufacturing Practice or Halal certificate. 6. Grow using food-based substrate. And the last one, organic and halal.
Ligno Biotech is one of the biochemistry industry. Thus, documentation is very important as one of their biochemistry principles in their company. In biochemistry, documentation typically refers to the recording, reporting of the experimental methods, result and observation. This is crucial for scientific reproducibility, peer review and communication of findings. Here are some key points related to documentation in biochemistry. First, notebook. Scientists need notebook to write down everything they do in the lab, what they use, the step they follow, and what happens. Next is the data record. Scientists will record the numbers and outcomes so the others can comprehend and carry out the experiment. Next, method. Methods need to be clearly stated, guidelines for doing experiment. This company, of course, emphasize the observations of their mushroom production. Thus, they will record any noteworthy from the process of the production. Next, to increase the confidence of the consumer, it's important to cite the sources of the concept or a data. Thus, the consumer can refer to articles or any review or research paper to get more information about this mushroom. Next is the ethic. Ligno Biotech find the mushroom in the jungle, thus they emphasize the ethic toward nature. Lastly is the troubleshooting, where the scientists documenting experiment issue and their solutions for their researchers can draw conclusion and improve what they are lacking. Next, the biochemistry principle is continuous improvement, where the implement of system for continuous improvement based on a feedback and result from quality control and customer feedback. In fact, Ligno Biotech use new technologies such as ozone sterilization, a process that uses ozone gas, a very effective method for disinfection, deodorizations and sterilization in various industries. These technologies is important for maintaining a clean and hygienic environment conducive to grow of the healthy mycelia and the mushroom of production. Thus, this technology will reduce the risk of contamination for the mushroom and avoid wastage of the raw materials. In conclusion, biochemistry is a behind-curve technology such as genetic engineering and medicines for improving our life and the environment. Thus, from creating new drugs to improving industry, biochemistry drives innovation and progress in various fields. That's all from Group 1 for biochemistry principles, and thank you for listening.